Hello, everyone. Can you all hear me? I hope so. Good. Can you just put your hands up to see that you guys can hear me? Good. Okay, perfect. Thank you. So I think we've got already some people coming in, but we will just start on time because it's lunch break. So let's be efficient. So hello, everyone. All the people coming in. Okay, so hello, everyone. Antena Foundation is pleased to welcome you all on our first webinar. My name is Renata Nogueira, and I will be coordinating this session. Okay, I'm a pharmacist and I worked my whole life in the scientific area. And since 2015, I was very lucky to join Internet Foundation, where I can use scientific resources to help the lives of unprivileged populations. And today, oops, sorry. Just learn how can I pass this. Okay, good. And today our agenda is going to be on scientific training and humanitarian cause. How can we combine them on the PhD? So among all these strategies that I learned while working in NGO environment to acquire high quality scientific data, this one, the cooperation with universities, is the one that I find most promising and with highest impact. Sorry, I'm hearing some noise. Do you hear? Okay. And by the time we finish this talk, you will understand why I think I'm so fond of cooperation with universities. So after my brief introduction, we will talk with Joëlle Hourier. She's already here, connected. So Joëlle did a PhD at the University of Geneva in pharmaceutical sciences and in the group of phytochemistry led by Professor Hofender. She's now doing her postdoc in the middle of the pandemic, but we are glad she's here in Switzerland. She's doing her postdoc at the University of North Carolina. So one fun fact about Joëlle is that she has a horrible mood when she's hungry. So I hope she ate something before coming here today. No, we are going to be very fast. And then after that, we will travel all the way to Germany, to the German side of Switzerland, where we will talk with Carla. So Carla, she has a bachelor in food sciences, and she joined the group of Professor Wolfram for her master and for PhD. And she's now doing her postdoc in the same group. And one fun fact about Carla is that she didn't know the Palau existed before she started the PhD. And if you have no idea where Palau is, please stay with us. You will learn a lot about this island. So after that, we will have some time for questions and concluding remarks, but don't wait until the end to start putting your questions in the chat box, okay? So Antena is a Swiss foundation based in Geneva. We are in the area for over 30 years now and present in many countries and with projects in the south. Let me just take a laser pointer here. Our innovative model is based on research, field testing, and dissemination. The idea is that our solutions are autonomous so that people in the field can use them. So they need to be affordable, they need to be efficient, and they need to be tested in the field. And that's what we use for all the areas that we work. So we are always present in the essential needs area, and we have several departments, but today we will focus only on the medicine unit. And one thing that I think it's very important that before we begin this discussion and why we're here and why do we need, for example, NGOs like Antena, why do we need to invest in research for unprivileged populations? So I guess we can answer this question with some scientific data. So this is a publication performed by Leiden University in 2019. And they wanted to confront the research effort with the disease burden. So what they did is that they divided the countries into income level, and they put the disease burden of each country in these categories. So for example, in high income countries, colon cancer is a burden. but not diarrhea, for example. So because we are very lucky that we have drinkable water in our houses. But if we take a look on low-income countries, diarrhea is a huge burden. And we have, it's actually the biggest cause of death for children under 2 years old. So it's very sad to notice that in some countries we have drinkable water in the house, and in other countries a burden disease is diarrhea, and kids die from it. And in order to assess the research effort on each type of those disease burden, what they did is that they divided the percentage of publication with the disease burden. And they also stratified this amount of publications into publications that have something to do with the pharma, like are authored or funded by the pharma, and publications that have nothing to do with the pharmaceutical area. So with the pharma companies. So what they found is that big pharma, they drive their research efforts on countries and disease burden that are more prevalent in high-income countries. So, for example, they are very happy to develop and put their research effort in a monoclonal antibody, for example, and they can patent it, and they can sell in the market this type of market here that pays a high price and have a good return over their investment. But on the other hand, they are less interested in, for example, finding a solution to curb diarrhea. For example, put their research effort on a water device, for example. So a water is a device that was adapted by Antenna Foundation that is possible to produce by water and salt, sodium hypochlorite, and then we can potabilize the water of many houses. And why are they not so interested? Well, because these people here, in order to buy a device like this, it needs to be very affordable. So what you see is that when the income of the country goes down, the interest of the big pharma goes down as well, which is kind of expected because it's a company. But what they found as well, which is very sad to notice, is that publications that don't have anything to do with pharmaceutical companies also follow the same trend. So if we take a look here, there is a huge research effort in disease burden that are more prevalent in high-income countries. So global health needs are unmet by research efforts, and that's a reality. But I invite you all to defy this and fight against this reality. What if we start collaborating between top-level universities, researchers from low-income countries, and NGOs that are aware of the needs from the field? Well, we will change this scenario. We will start having a lot of research effort on diseases that are a burden in low-income countries. And we will show you with the presentation of Carla and Joel that that's completely possible. But another point that is important to be discussed when we are talking about research for low-income countries is that this research needs to be adapted to the field. So for example, let's take a look at what is happening with non-communicable diseases. 67% of deaths in low-income countries are due to hypertension and diabetes, for example. And you can say, well, we have medicines to lower blood pressure. We have medicines to lower glucose in the blood. What is happening? Well, what's happening is that these research are not adapted to that situation. In order for a tool to be efficient to curb a disease in a country, it needs to be adapted to the conditions of the country. So it needs to be affordable. It needs to be as well as local, because they cannot rely on resources coming from outside. And also we need to work with strict collaboration with local NGOs so that the whole part of health literacy can be also made. So in my brief introduction here, what I would like to point some take-home messages that I think we can close this gap of global health needs. And I think that universities can play a key role on that. But the research that is conducted in this area needs to be more adapted so that they can be more efficient. Okay, and I will conclude with a phrase from our founder, Denis van der Weijden, that he said that we cannot change the world, but we can do what is on our reach. So I invite you all, from the people who are from university level, who have some power over your groups, to reflect on this possibility and reach out to NGOs so we can work together. I also invite you all from the audience to stay connected with us. We are present in different platforms. We have a website, a newsletter, and you can always subscribe to our channel. Okay, thank you very much. And I pass the word to Joël.
Thank you, Renata. Can you see my screen? And please interrupt me if you hear any problem of connection, because I cannot know and it's really stressful. So if you can give me some, yeah. So I'm really glad to be here, and I thank you, all of you, to take time to listen to this talk during your lunchtime. So my talk is entitled A Reverse Pharmacology Approach to Study Traditional Herbal Preparation. And yeah, I'm a pharmacist by training. As Renata said, I did my PhD in a group of phytochemistry. And today I'm a kind of postdoc in Professor Nadia Czech group. So I work remotely 2 days per week. And 4 days per week, I'm a kind of farmer in training in a farm in the canton of Jura. And with this farm, we have a project related to herbal preparation. When Renata asked me to talk about scientific training and
human... Just a second, you are not on presentation mode.
Oh.
Yes.
Is it okay now?
No, not yet.
Can we just stop sharing and start again?
Yes, good idea.
Yeah, our audience is so great. They are just giving several advices to us for the chat. Thank you, guys. Yeah, Mariana said full screen.
I am, but
maybe if you get out and stop, yeah, maybe you have to stop sharing and start again.
Okay. Is it better now?
Yes, perfect.
Perfect. So I start from there. And yeah, so yeah, Antenna Foundation invited me to speak about scientific training and humanitarian cause. How could I combine both on my PhD? And my first answer would have been to say, but it happened by chance. And then I took a few steps back, and I remember my deep motivation to start doing a PhD. And I think I can make a discussion about it, but instead of discussing this, I'm going to present you a project and open the discussion during the question and answer, and maybe focus on the humanitarian cause during the conclusion. So my main goal was to do scientific research related to herbal medicine. And herbal medicine contains herbal preparations that are mainly used by traditional and complementary medicine. And the World Health Organization reports that still a majority of the worldwide population use herbal preparation by choice or by necessity. And it considers that it is a potential source of affordable and effective treatment, but that must be evaluated in terms of efficacy, safety, and quality. And these are the three main pillars of all pharmaceutical sciences. When we speak about herbal preparation, we speak about a wide range of different products going from our infusion to herbal drug preparation and also botanical food supplement. And all these different products can be seen, that can be perceived as a source of bioactive natural products. And here we can do two different classes. On one hand, we have the herbs as a source of monosubstance. And I show you here two classic examples. We have the morphine, which is a major painkiller, and which is still extracted from the poppy, the Papaver somniferum, and even today. We have also the artemisinin, which was first isolated on this plant, Artemisia annua, and whose hemisynthetic derivatives are today used as antimalarial. On the other hand, we have herbal drug preparation that are an extract from the plant that contain many constituents. And two examples that are on the Swiss market are, for instance, the St. John's wort, which is indicated to treat mild depression. And a more recent example is an extract obtained from Petazites hybridus, which is indicated to treat allergic rhinitis. And what I want to say about this herbal preparation is that they contain many constituents. And from a scientific point of view, it may be very complex. We have potentially multiple active constituents that can reach multiple targets in our body. And all these constituents we may act together in a synergistic way or additive or even in an antagonist way. And this represents a high pharmacological complexity. And to study this, if we consider the classic drug discovery and development process, we're going to start with pure compounds that are going to be tested on a batch or series of preclinical studies, and only the most promising molecules are going to reach the stage of a clinical trial where the safety and efficacy of the molecule is assessed. And only after all these steps, the molecule can reach the real life world, so the population, and we make a difference between the efficacy of the drugs in a standardized clinical trial and compare, to the real world by using these two terms, efficacy and effectiveness. And all these steps lead to a validated drugs. When we speak about herbal drug preparation, to somehow, if we start with the same path, we have problem to evaluate this multi-constituent, multiple activity with in vitro. And a team of researchers suggested around 10 years ago to take the opposite way to study traditional practices. So it has to be used by the people first. We cannot do this for any kind of molecules that we take out of the nature. And so they call this approach the reverse pharmacology. And the first step is to observe, to study what happened in the population. And to do so, the goal is to do a survey, which is called the retrospective treatment outcome, which will analyze, which will do association, statistical correlation between symptoms and outcome, how the disease evolves. And the goal is to find sorry, I forgot to say also the treatment. So we have symptoms, we have treatments, and the goal is to observe if one treatment is always associated with an improvement of the symptoms. And this is my 1 minute of advertisement, because with the Antenna Foundation, we have launched such a survey in the frame of the COVID-19 pandemic. So, and really, I invite you all to participate to this survey. It's a kind of citizen science exercise, and that's it. So this is what we call the retrospective treatment outcome. And the goal is to select some herbal preparation to go directly on the clinical trial. I just want to remind that the clinical trial has the highest level of medical evidence, much higher than all the tests that are done in the lab. And once we have done these steps, the plant can enter in the lab to provide pharmacological evidence and to obtain hopefully a validated herbal drug preparation. What happened in the lab may be related to the development of methods to check the quality of the herbal preparation, because the plants are well known for their chemical variability. It's also to study what are the bioactive constituents, and depending on the plants, maybe one constituent will where the bioactivity and will be able to enter the classical path of drug discovery. So I did my PhD in a group of phytochemistry, so I focused on the analytical, on the chemical analysis of this herbal preparation. And here we have also a difference between the classic drug development, which start with one molecules. We have just one peaks, as you can see here. And when we start with herbal preparation, we have multiple peaks. And to study this, we use techniques that basically will separate the compounds that are present in an extract of plant, and we're going to detect them. And the techniques that we use to separate are called the liquid chromatography. So you can see here that one peaks is usually one constituent, and to detect them, we used mass spectrometry, which allow us to define the molecular formula of a molecule and to give an idea quite good of the chemical class. But we are not really able to be sure that it's exactly this structure. So we speak about putative identification or annotation. And when we want to make sure that it's really these compounds, we have to compare our data with a reference standard or to do purification, which require a lot amount of job and solvents and so on. So we try to focus first on this data, and then only when it's really needed, we go to these tedious steps of purification. So the aim of my thesis was to select herbal preparation by clinical efficacy criteria and to identify the active compounds that could explain the bioactivity observed in humans. To do this, as a phytochemist, I needed to work in a multidisciplinary approach. And we had on one side the biologist and on the other side, the medical team. And on the biologist side, we have the group of Professor Christian Wolfram with Carla here, and we really share our strengths for a project, for a couple of projects. And for the project I'm going to talk today, we had, with the medical side, a collaboration with the Antenna Foundation, and in particular with Bertrand Gras and Chris Quitalang. And they are both members of the Pacific Academic Institute for Research. It is not really well known here because it is located in the Republic of Palau, which is a tiny island here in the Pacific Ocean. And in this island, they have a high level of overweight and obesity. And Dr. Gras and Quitalang launched a project to study health traditions that could be helpful and culturally relevant around the overweight and obesity problems they faced. And they did a first retrospective treatment outcome study that highlighted the decoction of the leaves of this plant, this small tree, which is called Valeria nishidae, and that was promising for type 2 diabetes. Then they organized a clinical trial, which has a design randomized, double-blind and crossover as an adjuvant treatment for patients already under treatment but with unstable glycemia. And they observed positive results. And so these plants enter the lab, in both Carla's lab and mine, to evaluate this freeze-dried decoction directly in a diet-induced obesity mouse model. And we had promising results. And I really don't describe this result because Carla is going to speak about this in a minute. On my side, I checked the chemical, I started to do the chemical characterization of the plants. And we observed two main peaks. This one were mainly made of sugars, that is quite common in all plants. And the other one here was previously detected in this plant, and we could formally identify it as mangiferin, which was, unfortunately for us, really well described for its anti-hyperglycemic activity. So we were a little bit disappointed from our point of view first. So we've decided to check what are the smallest peak. And I could annotate some iriflophenone, also mangiferin derivative, and flavones. And among them, only two molecules had a few in vitro activity on one target related to diabetes, but it's not very predictive tests. And so we consider that there were enough constituents with a non-bioactivity to keep going this, to keep going the project. So we decided to do a fractionation strategy to test only the minor constituent without this mangiferin, or at least, without this mangiferin. So if we take the chromatogram of our plants, we generated three other fractions, one containing mangiferin and the other major compound, here an iriflophenone derivative, and two other fractions containing no mangiferin or very trace amount and all the minor constituents. We tested them proportionally to the extract directly into the mice, and this fraction 4 here was the most active one. So we were really happy because in addition to the mangiferin, which is well known, we could find other constituents that ameliorate the glucose metabolism. So then the next step was to really identify formally these molecules. So I did some purification of this minor constituent, was able to find 4 mangiferin derivatives, including 3 new structures that were never published before. And we could identify other flavone, including a new one for the botanical family. So from my point of view here, it's quite a global summary about this project on Valeria nishidae. We see that the decoction improves the glucose homeostasis in vivo, and that these results were consistent with the clinical study, and that the fraction of previously unknown minor constituent is mainly bioactive. So then it's time to go back to the first question: how can I combine scientific training and humanitarian cause? I have to say that, yes, maybe it happened a little bit by chance for me, but it happens because my big idea behind my thesis was to work on a reversed pharmacology workflow, and this makes the combination really easy. for me, it makes a lot of sense to work on a project that also makes sense in the local context of Palau, because it was launched by Chris Kitalong, who lives there and knows the people. And it was also good for me from not only the Palau project, but from really the main motivation behind to work on herbal preparation as affordable and effective source of treatment. I really want to participate to this kind of project. And I also put the local, and I put it in red because from our Western point of view, sometimes we have new plants reaching the market, but we don't always know what are the local conditions to prepare these plants. And I think it's one of the next points that I, from my point of view, want to add when I do research. I am sure that my question is, I want to make sure that the local resources are good if the plant is then used by a large public. And for this, I want to make a few acknowledgements. So this is a whole team for this project. We have the team of Professor Wolfram. Here is my professor, Jean-Luc Wolfender. Here is Bertrand Gras from the Antenna Foundation. And here is Chris Kitalong, who we were able to meet in Shanghai, in fact. And I thank you for your attention. And I just add one more time, please participate to the TO survey on COVID-19.
So thank you, Joël, for this presentation. And now, Carla, the floor is yours.
Thank you very much. So I'm also going to share my screen. Hope that works. Can you hear me, by the way? Oops. Just a second. Can you see it in full screen? Okay. So thank you, Renata, for inviting me to speak today and talk about my PhD project, where I looked at the anti-diabetic properties of this medicinal herb Joël just mentioned, the Valerianicidae leaves. And as also said, I joined the Laboratory of Translational Nutrition Biology at ETH in Zurich, which focuses a lot on the development and also function of adipose tissue, so fat tissue, and how it evolves and changes in the context of obesity. As Joël talked before me, some of the things I will say are a bit repetitive considering the introduction.
Sorry, just a second, Carla. Yeah, okay.
Sorry, I wanted to, anyways. Okay, so nowadays we don't only have the COVID pandemic, and I think it gets forgotten a bit at the moment that also obesity has become a real problem in the world. And if we look at these numbers, we can see that global obesity prevalence rates have also reached pandemic proportions during the last 50 years and almost quadrupled. And it's not only a phenomenon of developed countries, which have an affluent lifestyle, but also occurs in developing nations. And if we look a bit deeper and look into the, I just also make the laser pointer. If we look at the nations or at the ranking of obesity prevalence rates, we see that in both female and male obesity prevalence rate, island nations are basically leading. So it's 10 out of 10 island nations have the highest rate of obesity in females, and it's 8 countries regarding male prevalence. And for us, as Joël already mentioned, Palau, also Palau is in the statistics. It's on 5th place for female obesity prevalence rate and on the 3rd place already for male obesity prevalence in 2017. Unfortunately, obesity is not only a problem for increased body mass, which has direct effects such as impaired mobility or chronic kidney disease from the high body mass. It also comes with a variety of secondary complications which can eventually be lethal and heavily affect a life quality and expectancy, such as cardiovascular disease, or as of interest in my project, type 2 diabetes. And type 2 diabetes is defined by chronically elevated blood glucose levels, which on the other hand can lead to an additional array of comorbidities or damage to the body, such as nerve pain or the diabetic foot, which eventually leads to amputation, worst case. In addition, type 2 diabetes, these individuals suffering from the disease, they depend heavily on medication. So it's really also an issue for the healthcare systems. And the patients really depend on the drug for their life most of the time. And given the high prevalence rate of obesity in Palau, it's not surprising that also diabetes is very, very common. Almost 40% of both men and women in adulthood are in a state of pre-diabetes and are very, very likely to develop eventually diabetes. And already the percentage of people who have diagnosed diabetes is around 20%. And as Joel already mentioned, in 2015, the Republic of Palau declared a state of emergency on diabetes and other diseases related to obesity, and they initiated this reverse pharmacology approach to identify a herbal preparation or traditional medicine which is safe and also effective to use to treat hyperglycemia or elevated blood glucose. So this is what Joel just showed before, that in the survey performed on the island by Bertrand Grasse from the Antenna Foundation, they identified Valerian nisidae as the most frequently used to treat diabetes, and its use also correlated with the criterion of lower blood sugar levels. They then performed this clinical trial on the islands where the people drank the tea for 6 weeks, and they actually showed that the glycated hemoglobin levels shown here were significantly lower, and this is an indicator for long-term control of blood glucose levels. Also, the blood pressure went down upon treatment with the tea. And why is this also of interest in a wider sense? If we look at modern diabetes pharmacotherapy, or what we, let's say, use here to treat diabetes, many of the diabetes drugs on the market actually originate from natural products, such as a GLP-1 receptor agonist, or also the most sold anti-diabetic drug, which is metformin, originated in the early 20th century from the Galgea officinalis. And this is how my PhD came into the picture. So we wanted to know whether the extract works and what makes it work. And as well already outlined, it was a collaborative project, including the Republic of Palau, who gave the resources, the team in Geneva for the phytochemical profiling, and us here at ETH. I was by myself mostly performing all the in vivo experiments, and we also used various in vitro cell culture models to look at the mode of action and how in the cells the extract actually works, which mechanisms are activated. We were also able to identify which is the active compound within the plant, but due to time reasons and also our intellectual property interest, we will not talk about this at that point, or I will not. And so how did I test this in the mouse model? We came up with a mouse model that basically stimulates the development of obesity and type 2 diabetes as it occurs in the humans. So we put mice on a high caloric, high fat diet for 6 to 12 weeks, depending on the experiment. And then we started a dietary intervention for 7 weeks, where we allocated the mice to either just the high-fat diet food, or high-fat diet, which we supplemented with the valerianicidai extract. And as already shown by Joël, this was prepared in Geneva for us, and it was prepared in the traditional way as it's done in Palau. We then did a variety of metabolic tests to see whether the diabetes phenotype in the mouse can be improved. And in the insulin tolerance test, we give the mouse a dose of insulin and measure blood glucose levels over the time course. And insulin is the major hormone which stimulates glucose uptake after a meal. And as you can see, the mice which had the valerianicidai extract in the food, they reacted better to the insulin and could clear the blood glucose more effectively. We also did a similar test, the glucose tolerance test, where we give orally a certain dose of glucose and follow how the blood glucose is cleared from the bloodstream. And again, we could see that mice which were on the treatment more effectively cleared blood glucose levels after the oral glucose dose. So we really could also show that in our mouse model of obesity and diabetes, that the valerianicidai extract works. As we could see here that glucose is taken up into some tissues more rapidly than compared to the controls, we were wondering which tissues are actually affected. So we used a radioactive labeled glucose molecule. We injected it into the mouse, which was also either on high-fat diet or high-fat diet with the extract, and we harvested various tissue. We could not see any difference in glucose uptake between the muscle, in the muscle or the liver, but in different types of adipose tissue, so the fat. Especially in the white fat, we saw that the extract enhances glucose uptake and thereby reduces glucose load in the blood. As we saw that the effect seems to be specific for the fat tissue, we then thought we have a nice in vitro system, and we repeated the experiment where we treat fat cells for 3 days with the valerianicidai extract. And we could see again in the unstimulated condition, so if we don't add insulin in addition, the glucose uptake was significantly increased into the adipocytes. And we believe that the effect here we see in insulin is only an additive effect, as the basal glucose uptake rates are already elevated. Given this finding, we were curious what is the molecular mechanism of this higher glucose uptake we see in the fat in response to valerianicidai. And for this, I just need to introduce the way how adipocytes take up glucose. And this is done by 2 different proteins. So the adipocytes have the glucose transporter 1, which is responsible for basal and also insulin-independent glucose uptake. So it serves the basal requirements of the cell. There's a second transporter, it's called glucose transporter 4, which usually is within the cell, so it cannot take up glucose from the blood. But once it is stimulated with insulin, the cell, the transporter goes to the periphery and it can take up the glucose. So this is the insulin-sensitive glucose transporter. We checked now in our adipocytes after the valerianicidai treatment for the protein levels of these 2 transporters. And we found that especially the glucose transporter 1, so the one for the basal glucose uptake, was elevated. And we could also confirm this in the adipose tissue from the mice which were fed with the valerianicidai extract. And these results really go in line with the glucose uptake phenotype I have shown you on the previous slide. As Joël pointed out, she could identify that mangiferin is the major compound within the extract. And also on our side, we were kind of frustrated to notice that mangiferin is well known for its anti-diabetic properties, and there's a variety of well-performed studies showing its efficacy. So we received from Joël the 3 fractions from which 2 were depleted in mangiferin, and fraction 4 which still contained mangiferin, with the idea to identify minor compounds which have an additional benefit or might even confer the major part of the bioactivity of the valerianicidai extract. So we repeated our experiment. We again had the mice on high-fat diet and started the dietary intervention. And we measured then on course of the dietary intervention blood glucose levels at random time point. And especially over the longer term duration of the intervention, we could see that mainly fraction 4 lowers blood glucose levels. We also repeated the insulin tolerance test and the glucose tolerance test and could confirm that fraction 4 seems to be the most promising fraction regarding an improvement in glucose homeostasis. What we were curious then, whether one of the compounds which is found within fraction 4 can actually, or yeah, all the compounds together in fraction 4 can compete against a known anti-diabetic drug. So we prolonged the high-fat diet feeding period to 12 weeks to have the mouse more sick, so to have a more sick model. So we need to have stronger effect to improve the health state. And then we introduced metformin, the most common anti-diabetic drug, which we also added to the food. Also, the drug is taken orally. And we could confirm that after continuation of the dietary intervention, that both fraction 4, to a similar extent than metformin, could reduce blood glucose level. And this effect we could also confirm in the insulin tolerance test. So the effect size of fraction 4 is in an extent that it can actually compete to a known anti-diabetic drug. So as a short summary in my PhD, among many other experiments, which I don't have the time to show, we could show that valerianisidae improves glucose control in diabetic mice and that a subfraction of the extract can compete with a known anti-diabetic drug. Moreover, we could show how it works. So it increases glucose uptake into the adipose tissue by increasing the levels of this glucose transporter. And we were able to decipher the active compound within the extract. And coming back now to the question, what is the benefit basically of doing my PhD for both myself and also for the Republic of Palau, which is so heavily affected by diabetes and obesity, I could provide the evidence that the extract works in our mouse model, which confirms the human studies performed on the island. We could newly show what is the mode of action of the plant and also decipher which compound confers the bioactivity. And why is this important? If we think further that diabetes is a progressive disease, and already in Palau, people have treatment and also drugs to treat diabetes. But if there is an additional mode of action, we can add an additional layer of how blood glucose level can be improved in addition to the pathways that are already targeted by traditional medication, or not traditional, but the actual drugs. In addition, as we identified the active principle, one could also think whether there is a potential for Palau to commercialize the compound, which also would give a financial benefit. And in the end, all of them will end up in a nice scientific publication, which also raises awareness about what is happening in Palau. And as said, I was not even aware that this place exists before I started my PhD. And if we look for my side, I managed to get my PhD title. And I was able to use really state-of-the-art methods and facilities, which I think as a scientist is of your interest to do great work. And also a PhD can be frustrating, but it was nice to think that in the end, I don't work only for myself, but the project is part of a bigger picture and eventually the outcome might help people in need. I was able to work with different collaborators. I got insight into a new culture, which I was not aware before. I learned a lot also about phytochemistry, which is not my main topic. And I could be convinced that traditional medicine actually works and has a very promising direction as our active compound shows. On the other hand, of course, it's not always easy. I also would like to mention some points which should be kept in mind if you work in such a project. And first of all, you're a scientist, and as a PhD student, you want to create or find something novel. Also, it needs to be in your interest to get a good publication if you want like to stay in science and go for an academia career, depending on what you want. Also, you might have some diverging interests at one point, because together with that you want to find novelty, when we identified mangiferin, it was a bit going into a different direction where it's worth to look into the minor compounds. But as in our side, we were interested to find something new. We really pushed for this to actually do this fractionation approach. And in that sense, it's always important to clearly communicate and be transparent between all your collaborators. And you also sometimes need patience, because my mice maybe don't behave as they should, or Joel might be delayed in getting me the extract, which never happened, but my mice sometimes did not get fed as they should. And you also need to follow certain regulations if you work with genetic resources from other countries, and need to make sure that you follow Nagoya Protocol and have the required agreements to do clean science and have everything transparent. And with this, I would also basically show my acknowledgement, which is basically the same as Joel has. In addition, I would like to acknowledge the SNF and InnoSwiss, who now fund my postdoc year to finish up the project.
Thank you very much, Carla. I would like to thank from Antenna Foundation the opportunity that you both gave to us. So thank you too for being here. And I also thank both of your labs who accepted the challenge. So Professor Wolfender and Professor Wolfram, thank you very much, all the ones involved in this research. And I already have here some questions that I will read for you in the chat box. And people commenting, very nice presentations, thank you very much. What are the main problems to study herbal preparations, and how do you deal with plant variation compounds in the same plant species? So if you can please, I think it's Joël.
Yeah. Thank you for this question. So this is a wide question. I guess I start to answer the second one. How do you deal with plant variation compounds in the same plant species? I guess if we know the active compounds, it's quite easy. We can do quantification of the main active constituent. When there are more than one, it becomes more tricky, but today some quality control of the main constituents are possible, but the quality of the plants, if they come from different geographical, if they are collected at different time in the year, are one of the major issues in the field right now. It has to be said. And it also has to be said that if a herbal drug preparation is well checked, well controlled, we don't have this we are not sure of this when we speak about botanical food supplement, at least in the Western countries. And what are the main problems to study herbal preparation? If we consider what I presented, a reverse pharmacology workflow, the most tricky is to organize a clinical trial, because basically it's expensive, because it's not so, maybe we shouldn't say this as scientific, but it's not easy to find funding. And also because as it's a natural source, we cannot put any patent, so there is not a lot of industries that are interested to do this. And so these are on the side of the clinical trial. And then the second big category of problem is this multiple component that in an extract. So to face this, it's often it's somehow recommended to start directly in vivo, and in vivo may be a problem for ethics, or it's also very expensive. So yeah, I hope it answers the question, at least partially.
Perfect. Thank you very much. Here we have another question from Jan. Dr. Carla, please, what was the sample size for this study?
So for the animals, we had usually an N of 7 up to 10 for each treatment.
Good. Here we have, oh, that's so nice, because I am seeing here many people from my past life joining. Thank you all for participating. We have here a comment from Merlin Wilcox that, yeah, he says, you challenge universities to work with NGOs on problems of low middle income countries, but the problem is funding, and you already said that. The challenge is really the funders, and yeah, and in the UK, he says the global health funding has just been cut dramatically because of the impact of COVID. And also he has another question. Has anyone done a clinical trial of the Palau preparation in reverse pharmacology? Clinical trials come before in vitro pharmacology.
Yes, it was done.
Yes, keep along at all.
And yeah, it was done as an adjuvant treatment on 55 patients. And the next step would try to organize a clinical trial on, let's say, naive patients, just of people that should have to take now a new treatment that were in a pre-diabetes level, but it's under discussion.
Yes, it's on our list at Antena to do another larger clinical trial in Palau. And how I have questions from Vivian Letri or myself. It was clear how the science field lacks focus on problems related to unprivileged communities. What do you think that could be done in order to motivate and attract scientists, researchers to collaborate with humanitarian agencies, or simple research the basic needs and problems affecting these communities? Yes, as Merlin already mentioned, we have a problem of funding, but I also think that we have a problem of communication. So I think that on our side, at NGOs, we need to be more out there talking about what we are doing. And so please, if any one of you from the audience is organizing a symposium or conference, let us know and invite us. But I think I would say that there is a problem of funding, and there is a problem of connection. But another thing that the literatures bring to us is the problem of citation. Those diseases that are a burden on high-income countries, they receive more funding, but also when the work is published, they receive more citation, because you have the whole world trying to look for solutions. Also, there is the problem of patentability. What the base of our work is really to make this available. So we have several barriers, and I know that we won't going to have all the scientists in high and middle high income countries working in this area. But if we can just shift and redirect the priorities, like 20% of it, I think we will have a huge impact on low income countries. So I think, any more questions do we have? Here I have one question from someone from the team who asked, did you get to visit Palau? No, not yet. Yes, it's true, not yet. But
we were thinking about last year, but then Corona happened, so not happening.
Yes, I think it would be nice to go, all the team, to Palau. So I think, yeah, and here we have another question. Can you tell us more about the role of local academic partners, from Robert Blaise in the research?
I can answer. So Bertrand Gras, a medical doctor who works at the Antenna Foundation, spent, I think, 3 to 6 months in Palau during the RTO survey, I think. So he knows the people there, and he has a very strong collaboration with Chris Kitalong. And so he is the main, he is a person in charge of the communication mainly, but we had also a nice interaction with Chris Kitalong. It's a bit difficult because of the time zone. It's really different. But when you speak about the, when you ask about the role of local academic partners.
Yes, I think we also have another question. How closely did you interact with collaborators from Palau?
Yeah. With Chris, we had we did some updates about the results, and if we write him an email, he usually answers, so it's okay, I guess. And so for the first question, somehow Chris Kitalong is our partner, and he represents the academic in Palau. You have to know that this island have a population of 20,000 people, so it's really a small country. Maybe you want to add something, Carla? I don't know.
Mm-hmm. Great. Yes, I think that, well, it's already 1:30. And what I suggest that if anyone has another question for the group, please feel free to reach out to us. And we are very happy that we had on our first webinar at Antenna such a great audience, and we wish to do it more. And thank you all. Carla and Joel, you want to say something? Good. Yeah, thank you, Leticia Labriola. Oh my gosh, been so Robert Drummond, many interesting people participating. So thank you very much, and have a nice day, you all. Bye-bye.